I'm a very junior registrar. I'm an SD3, maybe an SD4 after my ARCP, but I've spent the last 10 years in genomic research. So I'll try to do 10 years in 10 minutes. I do get very enthusiastic, and you will lose me at a point, but please ask. I have warned my colleagues that I will ask them questions, but while this is setting up, final, I wanted to bring this here, because everybody's been sitting here for a few hours. I came in and it's very tame. So, before Paul and I are going to bore you with our stories, because the real excitement, as David said, is maybe not in the slides we show, but the excitement of doing science and all the pitfalls and the depressions. What do you want to know after 25 minutes? Do you want to know where the closest exit is? Do you want to know where you can get the coffee? Do you want to know when the bar opens? Or what do you want to know from Paul and us? Because there's nothing more dull than listening to slides and PowerPoint presentations. So, if you say nothing, we'll go for a walk. That's also okay. Okay, so I think the exits are down there and we'll start walking. Okay, so you've just given me carte blanche to talk what I want to talk about. But what I hope Paul and I can convey are a few things. So I just want to talk to you, there's a lot happening in genetics right now. There's a big hype and it will pass, but as long as it's there, it's interesting. So I just want to talk to you about what are we talking about? And some of the things I just want to talk to you about is, what is DNA again? Why is everybody hyping about it? Which techniques do we have to examine it, and why do we want to do this? And also Paul, more importantly, we'll be talking about ethical aspects and also aspects which we should be able to discuss about. So with my carte blanche, I will start right now. So I collect affiliations, so that's, you can see here. So what is it? DNA is deoxyribonucleic acid, and this DNA, you have long strands of it in our body. It's basically made, very simplified, of 4 building blocks, A, T, C, G. And we have, these are called base pairs, we have 3 million base pairs. This is all roughly, I know there are experts here, but just for you to imagine. And the exciting bit about all of this is that we only know relatively recently that this is there, like the last century. And the other thing we know is that only 2% of these 3 million base pairs are coding. So what does that mean? Only 2% of 3 million has a function in making genes and proteins. So the 98%, which we used to call, or the community used to call, junk DNA or desert DNA, is actually where the excitement is right now. So that's what you can see here. So you can see, DNA is organized in chromosomes, and they're all wind up together. So obviously it's not so beautiful as in the previous picture, and they're different areas. So there are areas where you have RNA made for proteins, and that's something you can investigate. That's like about the 2% I'm talking about. But there are a lot of other areas which regulate these bits. And in the last years, there have been a lot of fancy techniques which are listed here to look at all these regulatory elements. So that's what is actually happening right now to understand what these bits do. But just going back a little bit to the basics, I mean, okay, DNA, it's nice to have, but why? So the DNA in the nucleus is in a helix form, and there are bits which open up, and from that DNA, from a double strand, you open up and you make RNA, and this is all in the nucleus. This RNA Paul is an expert on that, is not immediately ready for production it has redundant bits in it, and it, like a lasso, it gets spliced into messenger RNA. That leaves the nucleus, and from the messenger RNA, that's kind of your code to make, in translation, make proteins, and they're then folded. So this is, we're talking about the 2% of the DNA. So if you want to, because I just wanted to talk about, you know, what's DNA, which techniques are we looking at. So we've now come down to one of the things DNA is useful for is to make proteins, and how proteins and genes, and how can you look at which genes are expressed in, for instance, a control sample versus an experimental sample. We say 10 years ago, we were like, oh, people with, say, an early MI, heart attacks, is that due to a specific gene expression profile they have? So we would basically take leukocytes from people who had a heart attack, people who didn't have a heart attack, extract their RNA, because that's where the message is conveyed for which proteins are made, reverse transcribe it, and then give a color to the RNA of the patient and another color to the RNA of the control, and hybridize them on these slides, which had probes for specific genes. Now we sequence them, but at that time they had probes. And then basically it was a show of lights. So if you can see that these plates would be scanned, and if you combine, if on a spot which represent a gene, to make it simple, if the patient had more of this gene expressed and you've labeled the patient red, the spot would turn red. If it was the control, and you'd labeled him green, it would become green. If they were both expressed, or expressed in both situations, then it would turn yellow. And with this information, you would assess gene expression. Nowadays we sequence, but this is still fairly done a lot. So gene expression arrays, and then this is something you will come across, or you might have come across already today. This is a heat map. And basically, the heat map, you can see in science we like nice colors and nice pictures, because the articles are too boring to read. And a heat map gives kind of an expression profile of which genes clustered are expressed more in one status versus the other. So gene expression is very interesting to see in what kind of state the cells are, it's a hype. Science, maybe I'm wrong, it's all about hypes. So we were, I think in 2005, 2006, doing a lot of expression arrays to obviously find the cause of disease, to cure people, and to become famous. But gene expression is still used, but we've moved on a bit more. And where have we moved to? We've actually moved to looking at variation in the genome itself. So that's another tool. So you can look at variants. And I hope you understand why you want to look at variants, because this variation in the DNA might cause disease. This is how DNA is replicated. So obviously, how do you get variation? How does it happen? Why are we all different? Sorry? No, go ahead. I think that was very good. I'm very happy that the question came from the Royal London. But I think we're different because our mums and dads are different, I hope, for most of us. And they're sometimes related. If you go back in the history of my family, they were all related because they were worried the money would go out of the family. So they're all very inbred. I'm convinced that that happens in UK culture too. If you go to a club in Chelsea, you're probably going to meet one of your second cousins without knowing. Anyway, so part of the variation comes obviously from the combination you get from your parents. So that's a reason to have variation. Except for having parts of your DNA and your variations from your father and your mother, in every individual, we think there are about 50 de novo mutations which can cause disease. There's obviously external noxes, so as radiation or certain chemicals which can cause variation. And things can spontaneously mutate, or with the process of DNA replication, which is safe because they're safeguarding moments, there can be variation. So I won't bore you with how the DNA replication goes, but variations is a hot topic. So this is it's a slide you'll see a lot if you go to talks about genetics, because what is plotted here is the frequency of these variations. So on the x-axis you see how frequent the variants are. So they can be very rare, rare, low frequent, or common. So we're talking about these variations in the DNA, and there's this hypothesis that depending on how rare, there's a correlation between the frequency of these variations and the effect size. Because you can imagine that if we all have rare variations, but if they were very powerful and had a very deleterious effect on us, that wouldn't fit. So we think that there are a few rare variants which have a high effect on a given phenotype. You can talk about anything you want. And there are more common variants which have a less effect size. And this is kind of the playing field for genomic research now and genetics. So if we go to these common variants with a low effect, that's what we were focusing on initially, and that's partly also because of technical advancements and the cost. So what we would do would be a very simple case control setting. So it's kind of what we did with gene expression. You would take a group of people who had disease X, so say I'm just making it simple, hemophilia A, then obviously we know what's going on there, and a group of controls which are matched. You would genotype them, and you would tag certain variants in that DNA which are representative of certain blocks, and we started with maybe 50,000 or even lower, and now you can type 1 million variants. You would then do quality controls, that's a lot of programming, and then you would associate. So you do chi-square tests to see is there an association between the variant and the disease, and those tests are actually relatively simple, and that because you need big numbers because of low effect sizes, it turned out to become meta-analysis reporting variants. So this is a recent paper in Cell from where I work in Cambridge when I'm not in London, and we looked at the common signals, or the signals underlying platelet, red blood cell, and white blood cell phenotypes. And here, this is a Manhattan plot. So this looks like the skyline of Manhattan, that's what they say. And you can see the chromosomes, you can walk through the chromosomes, and the p-value of these chi-square test and the association signals. And you do a very stringent control. But basically you can say there's a certain cutoff, so this yellow line is actually the cutoff, and these are all new significant signals. But these signals only give you locations of the genome with some association with the disease, but it doesn't say that they're causal at all. In the last 10 years, since the WTCC study came out, a lot of GWAS have been done, and there are like 6,900 loci associated with a specific disease, but that hasn't, in many cases, resulted in actually curing people, but just giving association signals. So if we're looking at specific variants you want to see, because the GWAS just looks at random variants, in clinical variants, if you know it's a specific variant which causes disease, the technique you would use is PCR. And in PCR you use the system of the human body, or a similar system where you have the DNA from the subject you want to study. You add a primer. The primer gives the location which you want to replicate, and with enzymes you replicate that bit to see if there are that specific variant or not. But if it's another variant, you will be able to detect it. Then after PCR, the golden standard to sequence these genomes, to actually look at every letter rather than specific variants, is Sanger sequencing. So in Sanger sequencing, again, you have a primer for a bit of the DNA, and then while you make the DNA with the DNA polymerase, you occasionally build in a kind of stop block. So normally, if I draw that, so this is actually quite useful to have, you would have a bit of, I do also make notes, a bit of the DNA you want to, so this is, can you guys see it? So DNA, and you have a primer, and you want to know which variant or sequence it is. So basically, what you do is no, see? Okay, good. So basically, you want to look at the variation of this. Yeah, we can move it forward. Paul, how am I doing for time? Okay, good.
Sorry, should I give you a hand?
So DNA. So basically, you want to know what the order of this is. So you have a few pots, okay, and DNA, a primer, and you have a polymerase. And these pots, just imagine these golden pots, have the building blocks to make, to replicate this DNA. So there'll be D and TP, so there'll be A's, let's put it that way, C's, G's, and T's, okay? But there'll be two kinds. One kind of A, which you can keep on building on, and there will be one kind of A where the reaction will stop, so you'll get a fragment. So there'll be, in this pot, different fragment size, but every time a stop A, to put it that way, is built in, you'll get a small fragment, but you know that at all these bits there is an A. You do the same for the C's, the G's, and the T's, and if you run that on a gel, I think I'm getting too complex now, you can basically follow the order at which each of these fragments were stopped to sequence a part of the genome. This is very laborious. It takes forever to do. So it is still the golden standard because this is the most precise way we can have it according to the literature. But we've moved on. And what we now do is what you might have heard of, and I was hoping you would ask for, is whole genome sequencing. So what you now do is you have this messy genomic DNA, you cut it into fragments, okay? So we're not talking about only those 2% which code, but you cut it in multiple fragments. You add linkers to these fragments, and does anyone know, how would you cut DNA? Okay, I've lost you. Anyway, you cut DNA by enzymes or bases. Then you get these fragments of DNAs, which you add linker proteins in to, and it's all done very on microchips. So with this DNA, with the linker proteins, you can run them you can put them on a flow cell, which has all these primers where to the linkers adhere. And there you can first duplicate or make more of this DNA and then start sequencing. And how does the sequencing work? So you have these kind of bits of DNA, you replicate, they come on to these primers which tag, with this tag, they kind of stick onto this flow cell. And then we like colors. What happens is there's a DNA production reaction going, and every time a specific nucleotide gets built in, a specific color is emitted, and by the change of colors in the growing strand, you can generate the full sequence, and we can do that for the whole genome within a few hours. So this might be too much. So if we've done a project where we've sequenced 10,000 people, and this is just basically what scientists like to do is show how big what they can do is. You can fill it out in any way, but basically doing those 10,000, we found a lot of variation. So there were over 55 million new variants we found, and they are divided through the genome in different ways. Anyway, a lot about DNA, but I think there's much more important stuff happening. I think there's a kind of a new French Revolution happening with Donald Trump and Brexit, so we might not be so comfortable anymore in science and things might change. But till then, this is what I wanted to tell you about genomic research. Please ask anything you want, and otherwise we'll go on to Paul.